Dear students, today we are going to see about the microscopic features of some of the endocrine glands, mainly pituitary, thyroid and parathyroid. So all the pictures are taken from D5 atlas. So what we are seeing here is the panoramic view of pituitary gland. So already in gross anatomy we have studied about it. So basically we have adenohypophysis and neurohypophysis. So adenohypophysis, once again it is having pars distalis, pars intermedia and pars tuberalis. So if we take neurohypophysis, that composed of pars nervosa, infundibulum and median eminence. So these are the basic components of pituitary gland, already we studied. So now we will see about the microscopic features. So if we see the outer aspect, there is a thin layer of connective tissue covering the entire pituitary like a capsule. It is very thin capsule. So underneath that we are able to make out that adenohypophysis and neurohypophysis. So in the next slide we will see in detail about the adenohypophysis and neurohypophysis. So in this picture we are able to make out that pars distalis of adenohypophysis. So this is the pars distalis part. So this is pars intermedia of adenohypophysis. So this is pars nervosa. So one by one we will see. So first, if we see that pars distalis part, we are able to make out cluster of endocrine cells. So we are able to make out many clusters of endocrine cells. So these endocrine cells are divided into two groups. So some of the cells what we are seeing here are darkly stained. So these are all darkly stained cells. So these darkly stained cells are called as chromophils. Some of the light stained cells we are able to make out here and there. They are called as chromophobe cells. So we have chromophils and chromophobe cells. So in addition to that, in between the clusters of cells, we have fenestrated capillaries. So of course that is the striking feature of any endocrine gland. So you will be having clusters or cords of endocrine cells. So in between the cells we will be having fenestrated capillaries. So already we know that endocrine glands means the secretions will be directly poured into the circulation. So that's why we are having a lot of fenestrated capillaries here. So now first we will see about the chromophils. So if you take the chromophils, once again they are divided into two groups, acidophils and basophils. So acidophils are otherwise called as alpha cells. Here if we see, some of the chromophils are darkly stained with eosin. So we have that secretory granules in these cells, they are stained with eosin. So those cells are called as acidophils or alpha cells. The other dark stained cells are stained with hematoxylin. Those cells are called as basophils or beta cells. So these are the two types of chromophils. Next, if we take the chromophobe cells, so they are light stained. So we will be having less secretory granules in that. Because of that it is light stained. So they are called as chromophobe cells, or otherwise they are considered as degranulated cells. Degranulated chromophils are called as chromophobe cells. So the main feature of pars distalis, so presence of the endocrine cell clusters. In each cluster we have chromophils and chromophobes. So these are all the chromophils. So they are acidophils, these are basophils. In between that we have chromophobes. So now we will see what are the functions of these cells. So before that, so this is the line diagram to show the features of chromophils and chromophobes. So now we see, so these are the acidophils. clearly we are able to make out that granules, so stained with eosin. So these are acidophils. Now these are all the basophils, so they are stained with hematoxylin. Clearly we are able to make out that secretory granules. Next, these are the chromophore cells. So in between we are able to make out that sinusoids are fenestrated capillaries. So this picture very clearly shows that the acidophils and basophils, of course chromophores also. So now what are the functions of chromophils? Chromophils, as I told already, we have acidophils and basophils. Once again, if you take that acidophils, those cells that secrete growth hormone are called as somatotrophs. So like that we have mammotrophs that secretes prolactin. So if you go to the basophils, we have thyrotrophs secreting thyroid stimulating hormone. So like that gonadotrophs we have secreting follicle stimulating hormone and luteinizing hormone. Next we have corticotrophs, so secreting ACTH. So the function of each glands already you studied in physiology. So you should be able to tell about the functions of these hormones what you studied in physiology. So now if we see pars intermedia, so this is your pars intermedia. So in the pars intermedia mainly we will be having cells stained with hematoxylin. So they are called as basophilic cells. So these are all the basophilic cells present in the pars intermedia. In addition to that, sometimes we can make out colloid cyst. So this is the colloid cyst with hematoxylin stained cells. So actually in human, the functions of these cells present in the pars intermedia unknown, but it is believed that so these cells are secreting that melanocyte stimulating hormone ACTH as well as lipotrophic hormones. So these are the features of pars intermedia. Next, if you go to the pars nervosa, so this is the pars nervosa. Here mainly we can make out the axons only. So these are all the eosin stained structures are the axons. So where we have the neurons. So already you studied in physiology. So hypothalamo-hypophyseal tract. So it is made up of the neurons present in the hypothalamus. So the axons are coming down to the pars nervosa. So what we are seeing in pars nervosa are the axons of the neurosecretory neurons present in the hypothalamus. So in pars nervosa we can make out the axons as well as axon terminals. So in that axon terminals we will be having storing of the hormones secreted by that hypothalamic nuclei. So that we will see here. So that hypothalamus where we have mainly supraoptic nucleus. So the neurosecretory neurons present in the supraoptic nuclei secretes vasopressin. So like that the neurosecretory neurons present in the paraventricular nucleus secretes oxytocin. So these are the hormones secreted by these neurons present in the supraoptic nucleus as well as paraventricular nucleus of the hypothalamus. So these hormones reach these axon terminals present in the pars nervosa. So mainly it is made up of axons of the neurosecretory neurons present in the hypothalamus. So in between the axons we are able to make out that fenestrated capillaries. So these hormones will be released into the circulation directly through these fenestrated capillaries. So in addition to this, what we are seeing here, you are able to make out the numerous nuclei. What are those nuclei? So these nuclei are the another cells present in the pars nervosa, that is pituitarians. So they are the pituitarians. The pituitarians are nothing but glial cells, supporting cells present in the pars nervosa. If you compare the population of axons and pituitarians will be less. We will be having more of axons only. So these pituitary are considered as supporting cells. So in the pars nervosa, we can make out the axons, axon terminals. In the axon terminals, the hormones secreted by that neurons will be there. Sometimes they accumulate to form a body called as herring body. Herring body we can make out in the pars nervosa. So these are the features of the pituitary gland. So the striking features. So in the adenohypophysis, we can make out clusters of endocrine cells. So once again they are divided into acidophils and basophils. Put together it is called as chromophils. Next chromopope cells. In between the cells we can make out the fenestrated capillaries. Next in pars intermedia, mainly we will be having cells stained with hematoxylin called as basophilic cells. Basophilic cells. Sometimes we can make out that follicles. So colloid cyst, or we used to tell as colloid cyst. Okay. So sometimes we can make out that. The main function of these cells are not known, but it is believed that they secrete melanocyte stimulating hormone, lipotrophic hormone and ACTH. In pars nervosa, numerous axons are there. Where we have the neurons are present in the hypothalamus. So neurosecretory neurons present in the hypothalamus. So the axons of those neurons pass via that infundibulum, then it reaches the pars nervosa. That only we used to call it as hypothalamo-hypophyseal tract. So that secretions, the hormones secreted by mainly supraoptic nucleus and paraventricular nucleus, so they reach the axons, then it reaches the axon terminals, where they are stored and released into the circulation through the capillaries present there, fenestrated capillaries. As I told already, in addition to that, we have the nuclei of pituitary. What are pituitary? Pituitary are glial cells. So these are the features of pituitary gland. So easily we can identify if you know these features. So this is the another line diagram to understand that pars nervosa or neurohypophysis. Here you see clearly you are able to make out numerous axons. So these are all the axons. So the nuclei what we are seeing here are nuclei of pituitary. So these are all the axons. Next what we are going to see is the thyroid gland. So already you studied about the thyroid gland in gross anatomy. So we are having the capsule covering the thyroid gland, that is true and false capsule. True capsule is the condensation of connective tissue. So from the true capsule, the connective tissue septa entering into the gland. So these are the connective tissue septa entering into the gland, dividing the your mass of the thyroid gland into small lobules. If you take each lobule, we are able to make out some spherical masses. So what are these masses? So these masses are called as thyroid follicles. So these are all called as thyroid follicles. So if you take each follicle, it will be lined by basal lamina. It will be lined by basal lamina. Over the basal lamina will be having cells. So these cells are called as follicular cells. So these are the follicular cells. So if you see the nature of these follicular cells, so here it is cuboidal. So sometimes it will be columnar. So what does it denote? So if the follicular cells are columnar in nature, that indicates that the thyroid gland is active. So in active stage, the cells, the follicular cells are columnar in nature. In inactive stage, they are cuboidal in nature. So here it is cuboidal. So like that we can make out numerous thyroid follicles, group of follicles easily we can make out. Then in between the group of follicles we can make out the connective tissue septa, where we can make out the capillaries. So these are all the capillaries. If you see in high power, if you see one follicle, that we will see in the next slide. So see that, so this is one follicle, so with the basal lamina. Actually this basal lamina will be made up of reticular fibers. In addition to that, we can make out the your lymphatic capillaries as well as your blood vessel capillaries we can make out here. So these are all the capillaries. Next, if you see inside, what is this? So we can make out one eosin stained mass. So in the previous slide also we can see in each thyroid follicle we can make out the eosin stained mass. So that is called as colloid. So this is colloid. So if you take one follicle, you will be having the basal lamina, then the follicular cells. Next, inside the lumen we can make out that colloid. So now what is the function of these follicular cells? So its main function is dropping of the iodide ions from the circulation. Then synthesis of thyroglobulin. So that thyroglobulin will be stored in the colloid. So that is the main function. So what are the various steps in synthesis and all, already you studied in physiology. So that is beyond the scope of this video. So whatever you studied in physiology, you should be able to recollect. If the examiners ask, you have to tell about it. So next important thing, in addition to the follicular cells, what are the other cells we have in the thyroid? Another cell is the parafollicular cells. Parafollicular cells, as its name indicates, parafollicular cells, usually that will present adjacent to the follicular cells, attached to the same basal lamina. So it will be attached to the basal lamina that will present in between the two follicular cells. Another but important point, that will not reach towards the lumen. So it will be very close to the basal lamina. So this is the actual site of parafollicular cells, but it will present in between the follicles also. So in this picture we are able to make out that parafollicular cells present in between the thyroid follicles. So these are the parafollicles, 2 to 3 or of course 4 parafollicular cells we are able to make out. So other name given for these parafollicular cells, C cells or clear cells. So the main function of these parafollicular cells, it secretes calcitonin. So the function of calcitonin already you know, it reduces the blood calcium level. So these are the points you should know regarding thyroid gland. So you should be able to make out that thyroid follicles. Easily you can make out that thyroid follicles. So in the thyroid follicles we can make out the follicular cells, nature of the follicular cells, next colloid. Okay, so at the center we can make out the colloid. So these are the identification point. First point, presence of thyroid follicles. Next, the cuboidal cells. Next, parafollicular cells we have to write, but in light microscope it is difficult to find out the parafollicular cells, but even then you can write. Then of course the capillaries. So next slide what we are going to see is the parathyroid. So in this picture we are able to make out thyroid also, thyroid along with parathyroid. So here you see very clearly, you can make out this part of this slide is the thyroid gland. So you are able to make out numerous colloid filled thyroid follicles. So these are all the thyroid follicles. So these are the follicular cells. So these are all the fenestrated capillaries. Now if we take that parathyroid, so this is the parathyroid. So in the parathyroid also mainly we have two groups of cells. The main cells present in the parathyroid, so these are the principal cells. Principal cells are chief cells. Principal cells are chief cells. So see, so these are all the chief cells. So these chief cells are smaller in size but more in number. You see, so numerous chief cells are there. In between here and there we are able to make out another group of cells. So they are called as occipital cells. So they are the occipital cells. So these are all the occipital cells. If you compare the occipital cells and your principal cells or chief cells, the staining character, if you see, it is light stained. Chief cells are light stained with eosin. If you see one cell, it is roughly polygonal in shape with central nucleus, but if you see the staining character, it will be light stained with eosin, light stained. Okay, but if you see the occipital cells, you see the cells are larger than the principal cells. If you see the staining character, it is deeply stained with eosin. It is deeply stained with eosin due to the presence of numerous mitochondria in it. So the main function, this one, still it is not known. Actually it is considered as resting phase cell of principal cells. Now what is the main function of principal cells? So it secretes parathormone or parathyroid hormone. So it increases the blood calcium level. So easily we can identify. It is difficult to take the separate section of parathyroid. That's why this slide is with thyroid gland. So we can make out the thyroid mass with the thyroid follicles. In addition to that, we can make out the parathyroid. So identification points for parathyroid: presence of chief cells or principal cells. They are more in number, small in size, with central nucleus, with light stained with eosin. The characteristic features of occipital cells: they are larger than the principal cells but less in number, deeply stained with eosin. Deeply stained with eosin. The function of these occipital cells, actually it is considered as resting phase cells of chief cells. So chief cells are secreting parathyroid hormone. So with these points we can easily identify that parathyroid. So in this PPT we have seen the characteristic features of pituitary, thyroid and parathyroid. So with these ideas, please go through that slides. So it will be easy for you. Easily you can identify these slides. Thank you, students.